Hello, everyone. Nice meeting you. I'm Ping Xie from Rutgers University in the United States. First of all, I would like to thank the organization community for inviting us to present our research work here. Today, the title of my talk is about the adaptor protein TRAF3 is an immune checkpoint that inhibits myeloid-derived suppressor cell expansion. As you know, cancer immunotherapies such as immune checkpoint inhibitors, CAR T cells, and cancer vaccines are a major breakthrough in cancer treatment. However, not all patients respond well to cancer immunotherapies, and this is due to immunosuppression found in the tumor microenvironment. And myeloid-derived suppressor cells are a major contributor of immunosuppression in tumor microenvironment. Myeloid-derived suppressor cells are a heterogeneous group of immature myeloid cells. They are crucial drivers of chronic inflammation and immunosuppression. Myeloid-derived suppressor cells can suppress the function of T cells and NK cells through a variety of mechanisms, and therefore they're recognized as barriers of cancer immunotherapy and also a prime target for cancer immunotherapy. We are studying one novel regulator of myeloid-derived suppressor cells, TRAF3. TRAF3 is a member of the TRAF family of adaptor proteins. It has a typical domain structure of a TRAF molecule with N-terminal E3 ligase domain and C-terminal TRAF homology domain. TRAF molecules are ubiquitously expressed in different cells of the immune system, and they're utilized by a number of immune receptors for signaling. To understand the function of TRAF3 in myeloid cells, we have generated and characterized myeloid cell-specific TRAF3 deficient mice. We found the specific deletion of TRAF3 from myeloid cells does not affect myeloid cell maturation or change the homeostasis of macrophages and neutrophils. However, when the mice are challenged with bacterial mimic or viral mimic, such as toll-like receptor 4 ligands or toll-like receptor 3 ligands, we found that these mice exhibit altered cytokine production profile, including almost abolished the antiviral cytokine type I interferon production, but greatly enhanced production of inflammatory cytokines such as interleukin-6 and interleukin-12, etc. Interestingly, when myeloid cell-specific TRAF3 deficient mice get older, they spontaneously develop chronic inflammation and tumors that often affect multiple organs. And we found this disease manifestation are often associated with expansion of CD11B and GR1 double positive cells in the spleen. The CD11B GR1 positive cells could be neutrophils, could be myeloid-derived suppressor cells. So we further characterize these cells by flow cytometry and by looking at their nuclear morphology using gamma cell staining. And we found these cells exhibit features of MDSC, including their immature myeloid cell morphology, nuclear morphology, like the ring shape and the oval shape, which is a typical morphology of granulocytic MDSC and monocytic MDSC. And most importantly, when we co-culture these CD11B GR1 double positive cells from aging mice with spontaneous chronic inflammation or tumor, they are able to potently suppress CD8 T cell proliferation induced by CD3 or CD3 plus CD28. So this demonstrates the immunosuppressive function of these CD11B GR1 double positive cells. And from this evidence together, we conclude these CD11-BGR1 double positive cells are indeed MDSC. So these are aging mice with spontaneous disease. We further studied whether these mice can undergo MDSA expansion in a model of chronic inflammation induced in young adult mice. So we use repeated injections of heat-killed BCG to induce chronic inflammation in young adult lethal control and local mice. And we found, again, the local mice exhibit aberrant expansion, doubled or tripled the percentage of MDSC, and also more than fourfold of increase of the cell number of MDSC in the spleen of the local mice after repeated injection of heat-killed BCG. So our evidence from both aging mice with spontaneous disease or young adult mice with induced chronic inflammation collectively indicate that TRAF3 is a novel regulator of MDSA expansion. Because MDSA can inhibit tumor immunity, so we employed a new model of tumor transplantation to study whether this is the case in young adult mice with chronic inflammation. So we use diffuse large B-cell lymphoma cell lines that are derived from aging TRAF3 knockout mice spontaneously develop diffuse large B-cell lymphoma. We use these cell lines to transplant young adult control and local mice. We found in the absence of BCG treatment, both genotypes of mice can actually resist tumor growth. They can reject the transplanted tumor. However, when these young adult mice were treated with repeated injection of heat-killed BCG first, and then transplant with the tumor, these mice now have impaired tumor immunity. About 30% of the wild type mice cannot reject the transplanted tumor. In contrast, almost all the local mice cannot reject the transplanted tumor, and the tumor growth cause early death of these transplanted mice. And we found the knockout mice not only have increased growth of the transplanted tumor at the tumor inoculation site, there's also metastasis of the transplanted diffuse large B-cell lymphoma to lymph nodes, to the liver, to the spleen, or the chest or peritoneal cavity. And we also showed the statistical data of tumor weight, number of tumor, and the instance of metastasis in these graphs. And we also verified greatly expanded MDSC population in the spleen and also in the tumor microenvironment of the transplanted mice that received repeated BCG injections. So together, these results indicate that TRAF3 expressed in myeloid cells can promote tumor immunity indirectly by controlling MDSC expansion. We then try to investigate how MDSC hyperexpansion can actually affect tumor immunity. We found MDSC expansion can actually downregulate CD62L, also called L selectin, a lone target of MDSC expressed on T cells and NK cells. And we verify this in both CD8 T cells, CD4 T cells, and NK cells. In addition, we found that MDSC hyperexpansion can also affect the expression of two immunoinhibitory receptors, CD38 and KRG1 on CD8, CD4 T cells, as well as NK cells in transplanted MTRAF3 knockout mice with chronic inflammation. You can see the percentage of CD8 positive T cells is greatly increased in the local animals. So impaired tumor immunity is also mediated by downregulation of CD62L and upregulation of CD38 and KRAS1 on T cells and NK cells in the local mice. So our next question is how could TRAF3 regulate MDSA expansion? Because understanding the underlying mechanism may reveal new therapeutic target to inhibit MDSA expansion in cancer patients. So we use mixed bone marrow chimera experiment to answer this question. We use the mixed bone marrow from the TRAF3 knockout, which is a CD45 2 positive, and Y type, which is 45-1 and 45-2 heterozygote. We mix them in 1-to-1 ratio and adoptively transfer these mixed bone marrow cells into irradiated recipient mice, SJL mice that are CD45-1 positive. And 7 weeks after the transplantation, we check the chimerism by flow cytometry of blood cells, and then we provide 3 repeated BCG injections to induce chronic inflammation. And if our results show that the ratio of knockout to Y-type MDSC is higher than 2, then the mechanisms will be cell intrinsic. If the ratio is 1, then the mechanism will be cell extrinsic. If the ratio is between 1 to 2, then both intrinsic and extrinsic mechanisms contribute to TRAF3 mediated regulation of MDSA expansion. And our results of the mixed bone marrow chimeras show that the ratio of CD11B-GR1 in the spleen and bone marrow between the local to the Y-type donor bone marrow cells is about 1.5-fold. In contrast, we did not see such difference in the ratio of knockout versus Y-type B cells or T cells. So from this data, we conclude that both extrinsic and intrinsic mechanisms contribute to TRAF3 mediated regulation of MDSA expansion. To identify the extrinsic mediators that can contribute to TRAF3 mediated regulation of MDSA expansion, we performed cytokine chemokine protein array, and we identified 2 molecules, GCSF and the chemokines CXR13, are consistently elevated in both aging TRAF3 knockout mice with spontaneous tumor and chronic inflammation, as well as young adult mice that are induced with repeated injection of head-kit BCG. So we performed a neutralization experiment to identify which of these 2 molecules are the major driver of MDSA hyperexpansion in the local mice. And our results indicated that it's GCSF but not CXR13 that drive MDSA hyperexpansion in the local mice. We then sought to identify what cells can produce GCSF, and our data shows that it's inflammatory monocytes in the blood and also inflammatory macrophages in the spleen or other tissues of the knockout mice that can contribute to the elevated level of GCSF in blood circulation. So although the percentage of the GCSF producing cells is markedly increased in the local mice, however, the GCSF production is not different on a per cell basis in the 2 genotypes of inflammatory monocytes or macrophages. So we next sought to understand why we observed the increased percentage of inflammatory monocytes in the blood and also increased percentage of inflammatory macrophages in the spleen. We test the hypothesis that maybe these inflammatory cells express higher levels of chemokine receptors in the local mice. So we examined the expression level of different chemokine receptors of monocytes and macrophages, including CCR2, CXCR3-1, and CCR4. And we found that the G-CSF-producing inflammatory monocytes are CCR2 low, CXCR1 low, and CCR4 positive. However, again, we did not see difference in the expression level of all these chemokine receptors between the two genotypes of inflammatory monocytes or macrophages. And we also demonstrate the bacterial molecule LPS, the ligand for toll-like receptor 4, can actually downregulate expression of CCR2 and CXCR1, but upregulate the expression of CCR4 on monocytes in cultured bone marrow cells. However, again, we did not detect difference in the expression level of these chemokine receptors between the two genotypes of monocytes. So we next tested an alternative hypothesis that it's the chemokine ligand of CCR4 may be different between the two genotypes of mice. And we found that this is indeed the case. The ligand for CCR4, CCR22 level in the blood is highly increased in the local mice after both 2 hit-killed BCG injections as well as 3 injections of hit-killed BCG. And then we try to say whether the increased level of CCR22 is contributed to by increased production of this chemokine by TRAF3 knockout macrophages. So we use cultured bone marrow-derived macrophages to address this possibility, and we found indeed TRAF3 knockout macrophages produce striking increased levels of CCR22 on a per cell basis in cultured macrophages in response to stimulation by the bacterial ligands LPS. So in summary, in terms of the cell extrinsic mechanisms, we found that toll-like receptor ligands derived from bacteria, such as LPS derived from bacteria, can engage toll-like receptor signaling to stimulate production of chemokine CCR22, which will attract the CCR4 expressing inflammatory monocytes from bone marrow to blood circulation. And these inflammatory monocytes in turn produce G-CSF, which can recruit the GM progenitor cells from the bone marrow to blood circulation, and these progenitor cells can develop into MDSC. And meanwhile, the inflammatory monocytes can also migrate to the spleen and other tissues to differentiate into macrophages, which can also produce both the chemokine CCR22 and G-CSF in response to bacterial ligands, the toll-like receptor ligands. So these chemokines and cytokines can further amplify this feed-forward loop and induce MDSC expansion in the spleen, and we also detect it in the liver and blood. In the presence of TRAF3, TRAF3 can inhibit CCL22 production, which can inhibit the recruitment of inflammatory monocytes to produce G-CSF, and TRAF3 can also inhibit both CCR22 and G-CSF production in inflammatory macrophages in the spleen. So therefore, TRAF3 can inhibit MDSC expansion during chronic inflammation. We also investigated the intrinsic mechanisms of TRAF3-mediated regulation of MDSC expansion. We first found that GM-CSF-induced proliferation of CD11BGR1 double positive cells is most strikingly affected by TRAF3 deficiency. We then studied the early signal events of GM-CSF and found that the phosphorylation of STAT3, but not the phosphorylation of STAT5 or ERK1/2 induced by GM-CSF, is actually affected by TRAF3 in CD11BGR1 double positive bone marrow cells. So TRAF3 specifically inhibits GM-CSF-induced STAT3 phosphorylation in the progenitor bone marrow cells. And we verified the TRAF3 mediated regulation of GM-CSF-induced STAT3 phosphorylation using splenic MDSC purified from BCG-treated knockout mice. You can see the elevated STAT3 phosphorylation in the local splenic MDSC compared to the linear control wild-type MDSC. But the STAT5 phosphorylation and ERK phosphorylation is not affected by TRAF3 deficiency. So together, our evidence indicates that TRAF3 specifically inhibited GM-CSF-induced phosphorylation of STAT3 in both CD11BGR1 double positive bone marrow progenitor cells and splenic MDSC. And this pathway is targetable by STAT3 and JAK inhibitors. So these inhibitors can actually abolish GM-CSF-induced proliferation of MDSC in both genotype of mice. And then we performed quite extensive co-immunoprecipitation experiments to understand how TRAF3 can affect GM-CSF-induced STAT3 phosphorylation. We found that TRAF3 can facilitate the association of STAT3 with the phosphatase PTP1B, which can dephosphorylate STAT3. So in summary, we found TRAF3 does not affect GM-CSF-induced STAT5, AKT, and ERK activation. However, TRAF3 specifically inhibited GM-CSF-induced STAT3 phosphorylation by recruiting the phosphatase PTP1B to dysphosphorylate STAT3. Therefore, TRAF3 can inhibit GM-CSF-induced proliferation of CD11BGR1 positive bone marrow progenitor cells and splenic MDSC. So in summary, our study has elucidated one toll-like receptor TRAF3 CCL22 CCL4 GCSF signaling axis that is acting in inflammatory macrophages and monocytes, and also another GM-CSF TRAF3 STAT3 PTP1B signaling axis that is acting in MDSC. And these two pathways act so extrinsically and so intrinsically to coordinately control MDSC expansion during chronic inflammation. With that, I would like to take this opportunity to thank my lab members, my collaborators, and our funding support. Thank you for your attention. If you have any questions, you are welcome to email me, and I would be happy to address your questions. Thank you. Bye.